which is Applied Statistics. And my name's Phoebe Fitzpatrick. I'll be chairing this session. And first up, we have Amon from the Christchurch Hub, who's presenting on clustering approaches for linking large administrative data sets. So take it away, Amon.
Thanks, Phoebe. And before I get started, thank you to the organising committee. They've put in a lot of hard work getting the conference going. So I'm Eamon. I'm from the Australian Bureau of Statistics, where I work a lot on data linkage problems. So a little bit more context on that. At the ABS, we often get administrative data sets from other government agencies. We'd like to merge these together, but there's not always a key that we can merge them on. So what we do instead is we rely on identifying information like names, addresses, dates of birth, etc. And we use these to make our educated guess of which records in the different data sets refer to the same person. Here's some brief information about the type of data we have. And sort of, you can visualize this as overlapping data sets where there's not perfect overlap, but we can still link many people between them. One thing to keep in mind is that the data sets contain duplicate records. So not only do we have to find people who are the same across data sets, but also within the same data set. Here's a quick example. So I'm going to focus on clustering for this presentation. The process of clustering is grouping together the records who we think refer to the same person. And the step before this that I'm going to take as a given is a type of measure of similarity between pairs of records. So we can think of this as a probability that they refer to the same person. So we have a few people here. How many distinct people do we think are represented here? Someone from the audience. 4. Zach thinks 4. All right, 3. I'm going to go with 3. So my thought is John Smith is one person. George Smith might be his twin. Sarah Johnson and Sarah Black are likely the same person, but with a name change. So how do we cluster these? Here's our first idea. We call this connected components clustering. So if we only look at the edges with a match probability of 0.95 or higher, these are the edges that we keep. And if you think about it as a graph and take the connected components, these are the clusters that we end up with. So probably this is what Zach had in mind, separating Sarah Johnson from Sarah Black. We get these 4 clusters. But we've lost that link between, on the right-hand side, between Sarah Johnson and Sarah Black. If we reduce the threshold to something a bit lower, we only get 2 clusters. So we start to link together inappropriately those 2 clusters on the left, who are likely to be twins. So our model for assessing the similarity has some weaknesses. There are some real-world effects that mean that sometimes we overestimate the match probability for certain types of relationships. Okay, so what else can we do? One other approach is what we call single best links. So this is from one data set, meaning the records on the top, to the second data set, meaning the records on the bottom. Each record can only link to a record on the other data set, which is its best match. So it's fairly simple criteria. There's a few different ways of doing it. If you're interested in those methods, I've referenced a paper there, the bipartite matching algorithms. Give it a read if you're interested. So in this case, we would have George Smith, John Smith, and Sarah Johnson and Sarah Black would all be linked together. But that duplicate record for Sarah would be lost. That would be put in a separate cluster. So here's the idea that I put together for what we're working on. It's very simple. Doing it in 2 stages. First, where we use connected components with a higher threshold, and the second stage where we use single best links at a lower threshold. So this is what the first stage would look like. A high threshold of 0.95, we pick up that duplicate link for Sarah. And the second stage, that extra link is found because that's the single best link to the other data set. The single best link to a cluster on the other data set. Okay, that's all. Thank you very much. If you have any questions, contact me there.
Thanks, Amon. And next up, we have the Christchurch Hub again. And It is Soyun, who's investigating the effect of repeated patients in surgical outcomes research.
Kia ora, everyone. My name is Soyun Annie Park, and yeah, I'll just start off by briefly going through how this project came to be. Oh, not loading. Okay, so a bit of a background. So this project all began with my supervisor's initial paper comparing Auckland Hospital's performance before and after the implementation of the WHO surgical safety checklist. And a part of study involved comparing mortality rates across different time points. And he noticed a bit of a discrepancy in the mortality rates depending on the operation selection method. And this is where I picked this project up. And I couldn't really grasp what the problem was initially because my understanding of mortality rates from textbooks and lecture slides was simply as counting the number of new deaths and dividing it by the number of total patients within the given time frame. But unfortunately, it's not as simple as that. So imagine a situation where there are patients who receive more than one operation within a given time frame. So, for example, the data setup that I was using had over 90,000 operations, and these operations were received by fewer than 60,000 unique patients. So, if this is the case, which number do we use as our denominator? Is it going to be the number of operations, or is it the number of unique patients? So that's the question that I had. And I got curious and I did a little scoping review going through all the studies that has been published within the past 12 months that reported on surgical outcomes, specifically on the mortality rates. And the majority of the studies actually did not mention how they handle repeat patients or they even had any repeat patients at all. And for the ones that did, there was quite a variety of options there. Some studies included just the first operation, some included the very last operation, and some included just all of them, and some just did a random selection per each unique patient. And a number of studies also reported that the data set they were using was just completely de-identified. So they couldn't even tell whether they had any duplicate patients in their data set. So this is my research question. Because there are such a variety of approaches handling these repeat patients, will the measured mortality rate vary depending on the operation selection method? If so, by how much? So to do this, I looked at a moving average mortality rate across different operation selection methods. And I noticed there is a discrepancy of up to 1.5 percentage points. And this number might seem initially very small, but imagine a cohort of over 100,000 patients. This number translates to up to 1,500 deaths, and which is a substantial difference, significant difference in the context of surgical outcome studies. And people might say, what if we just choose one of those methods and use it consistently? Problem solved. But it's actually not the case because I looked at the moving average mortality rate across different window sizes, across different operation selection methods, and you can also see that the impact of the selection method isn't consistent across the study timeframe. So even if, say, everyone uses the first operation only method, the results might still vary depending on the duration of the data set they have. So in conclusion, capturing the true mortality rate isn't as easy as we think. So I'll be continuing my research in this regard until I submit my thesis next year. But one thing seems very clear. So we need a standardized protocol for measuring these mortality rates to ensure reliability and comparability across different surgical outcome studies. And hopefully it will lead to better insights and better patient outcomes. Thank you.
Thanks, Soyun. Next up is Xi Zhang, also at the Christchurch Hub, and they'll be presenting on a ruler detection method for auto-adjusting scales of shoe print images.
Thank you. Hey, my name is Jason. I'm a data scientist at the Institute of Environmental Science and Research. And my work mainly interested in using statistical and machine learning method to solve the problem in forensic science. And today I'd like to talk about part of my on the automated ruler detection method for adjusting the image scales. Okay. Yeah, the scale inconsistent image scale is often advised in the interpretation of forensic image comparison, especially in shoe print images. And so here's the example of it. We have the two shoe print where I capture in the lab, and we virtually see that without any further information, we will see the right one is greater than the left one because it's how our eye looks like. But in fact, they are made by the same shoe. And the reason for this is because we use different camera with different resolution, and the distance between the camera to the shoe print is inconsistent. And the way that we solve this problem, forensic solve this problem, is to place the ruler beside the shoe print. Therefore, we can use the information from the ruler to help us to get the scale of the images so that we can resize it to still make the comparison consistently. And however, this process has been conducted manually, and therefore we made a method to automate this process. So here's our method where we take the images as input, and first we put the images into a thing called the ruler region extraction block, which means we're only interested in the ruler region here. And then we employ a method called the iterated ruler line detection, which is roughly speaking, it just draw a line perpendicular across all the tick mark on the ruler. And here's the algorithm that shows in here. And if you're interested in, there's a little publication shows in the column. And then once the line has been detected, we apply a method called the ultra thresholding to convert the images into a binary images so that we can then extract the pixel along the ruler line. Once we're doing this and we plot sequentially, we can obtain a sequential plot that shows in here. And how the scale has been estimated, we just basically count how many pixels within each oscillation and then finally take the average of it. And here, just a little bit detail of how this method on getting the ruler out. So we use a thing called the image segmentation. So how it basically works is just that it takes the cat as images as input to the model, which we use the segment NSA model here, which proposed by the Meta people last year. So how this model works is just compute other binary images with one that tells where the cat sit in, and it's 0, that means it's other thing else. So here just the background. So we use this method to we initially use this method to our ruler, but it doesn't work because the method the model haven't seen the many ruler before. Therefore, we use a fine-tuning process to adapt this knowledge to the model. So once we're doing this, we can use the inference stage by taking the image back to the model, and then we output the binary images that tells us where the ruler sit on. Yeah, so here's some result. We can see that we have actual pixel per millimeter and the estimate per millimeter. We can see that there's still a little bit error, but might be in acceptable range. And the plot source on the y side is just the plot the ground truth on the x-axis and the estimation on the y-axis, and just apply to all the data set we have. And you can see they're aligned to the 45-degree line, not too bad thing. So we're still doing some not horrible work, and the average error here is about 3-4%. So yeah, here's the end of my talk. Thank you.
Thanks, Jason. Next up at the Hobart Hub, we have Shamali, who's going to present on advancing dynamic point processes modeling in ecology.
Thank you, and hello everyone. I'm Shamali Sujiva Kumari. I'm from RMIT University. I'm a PhD student. So my research is about advancing dynamic point process modeling in ecology. Then imagine you are standing in a rainforest with the surrounded towering trees and with the vibrant biodiversity. However, this ecosystem is constantly changing. So it is essential to understanding these ecosystems to monitor the ecosystem health and the predict responses to the environmental changes. Then through my research, what I am going to do is I'm trying to identify this variation, and I'm trying to give a mathematical framework for this ecological scenario. And climate change and habitat loss are considered as the main influential factors for these variations. Now, identifying complex species interaction and spatial and temporal variations are the major two challenges in my study. And also there are limited accessible tools to this type of analysis. So in my research, I am using dynamic point process model, and also I apply this model to the real world data to check the model performance. As well as finally, I will develop a user-friendly application to do this analysis easily without having much knowledge about statistical tools. So those are the two literatures that I am following mainly. The first literature, they have modeled their interaction by considering the neighboring objects only, left-hand side neighboring objects. The second literature, the very recent one released on this year, in this study, they have modeled the interaction, but that process interaction by considering the number of species. As I explained earlier, to check the performance of my proposed model, I am using real data set. This is the data set which I am using for that purpose. This is a open source data set which is collected from the North Queensland, Australia. As you can see here, there are 20 permanent rainforest plots. But in this photograph, you can see example of 3 plots. You can see here there are vibrant species available in these plots. So in this graph shows the distribution of the species in the Downfall Creek plot. Yeah, this is how I implemented my spatial birth date process. Here, each tree I consider as the individual point, and I'm proposing a new birth and death rate function by considering the spatial structure. That is the key difference with the previous literatures. In my research project, I am taking the interaction effect by considering the distance among all neighboring trees within the threshold distance. And here you can see for the birth and death rates, there are 2 parameters, alpha and mu, which is those 2 parameters that I need to estimate it through my simulation process. And those are the 2 maximum likelihood estimations that I have proposed for the continuous time and the discrete time separately. To propose this maximum likelihood estimation, here for the continuous case, I have considered 3 probability terms. The first one is the probability of waiting time. I consider it as exponentially distributed, and the second term is the probability of the type of event, and third term the probability of specific location. The birth location and the death location likewise. And this is the simplified likelihood estimation, likelihood function for the continuous time case. And similarly, I have proposed the likelihood function for the discrete time as well. Here also I am considering 3 probability terms. The first one is likelihood of observing n events, and the second term likelihood of observing NB births and ND deaths. For this purpose, I am using binomial distribution. And the last term is the probability of specific locations, as similar to the previous continuous time case. These two are the newly proposed likelihood functions for the parameter estimations. And these are the results that I have obtained from the simulation. Here I did it for the 50 replication and for each trajectories. As you can see here, the likelihood functions performs the results well. And you can see when we increase the number of trajectories, it shows the good estimation results. And up to now, I have implemented the simulation program for the single species and did the model validation using the real data. And further, I'm planning to expand this to multiple species and incorporate the environmental effects for this. And finally, I will develop the user-friendly application. Those are the references that I have used. Thank you very much for participating.
Thanks, Shamali. That was great. Next up at the Hobart Hub, we have Nur, who's going to be presenting on investigating students' performances in mathematics through PISA using machine learning. So take it away, Nur.
Oh, sorry. Good afternoon, everyone. Would that be okay? Before we start, I just want to ask you, how many of you consider that your parents influence your math performance score at high school? 1, 2, 3. All right. So online participants, there are 1,000 say yes in this in-person hub, and I believe that there are more than 1,000 saying yes at the other place. So let's see whether we have that answer. So this is our preliminary research that we have done, but this is only an initial research. That's why we put that in an abstract. We work with Mali, my college at Ramati, and my other college at the Indonesian University. So we are investigating the students' performance, particularly for math, through the PISA using machine learnings. So right, so let's see if I can see. All right, so what is PISA? So PISA is the International Student Assessment. It's being held by the organizations Economic and I can't remember, Corporation Developments based in UK. Every 3 years they run these assessments, and then nowadays there are 79 countries who has been involved, including Australia. Australia has been involved in this PISA in the last 2 rounds. So we take some, they took some score, particularly on math, science, and readings for 15 years old students. So it's about like a year 10 in Australia. So particularly in math, we're looking at we examine ability to solve the math, problems using their knowledge, and then how we implement that on the real life situations. It is more about problem solving, applying math concepts, and how they apply in the real world situations. So it's a bit different with assessment that we have in Australia, which is NAPLAN. So now we are wondering, this one is just a bit, a quick information. In the last 2 runs, 2018, it's very small there, but Australia lies on the rank 21 there, and then the last one there on 2022, Australia went up into 11th. So that is very interesting information there. And then, and we're just wondering what then, we want to understand what works, what factors that is impacting this student performance, especially in math. So by understanding this concept, what factors that are involving in the student performance, maybe then we can give some interventions to improve the performance of the students. maybe but because maybe we can then improve the learning environments and then focusing on more on the disadvantaged background, particularly in more under development countries, etc. and etc. And then so the research here is we want the initial one is we want to investigate the relationships between some of the factors there with the student math performance. We are using this PISA data, and then we want to look at what is the most important features. Then maybe then after that we can then improve what are the performance through these factors. Later on, we want then to further analyze how do we then can we predict the scores by using the machine learning, but we haven't done this one first. So about PISA data set, there are 27 futures. So these are the futures. They are grouped by the student background. Some of them are the parental backgrounds that I just asked you at the beginning. Technology, school policies, the learning environment, and so on and so on. Yeah, so the machine learning that we use, there are about 5 there. And then, but the justifications, we believe that these models are good enough to handle the data, and there's the strength, the weakness, but I'm just going to skip this. Of course, there are some data pre-processing. We also, each of the algorithms, they have the key metrics, but then we're looking at them, just to summarize, we're looking at this preliminary result, and then we end up that XGBoost has a good result, the best result for amongst those 5. Then this is the most important one for our initial result, that the top predicts of the performance. The first one is actually the parental education level. The second one, and the school resources, access to digital services, and teacher-student ratio. So for conclusions, this is initial conclusions that parental education backgrounds play important roles in student performance, especially in math. But however, we have a further questions. Why? How? What about the others? Because my own belief that teaching practices also play important roles. And this is what our further research is going to be. And thank you so much for listening.
Thank you, No, that was great. Our final presentation is also in the Hobart Hub, and Hub 2 is going to be presenting on simulation of conservation management strategies.
Okay, thank you for the introduction. And yeah, my title is also introduced. It's on simulation of conservation management strategies. And I'm working this closely with my supervisors, Barbara and Malgorzata. Yeah, before I talk about what's genetic rescue, I'll try to first why conservation matters. Yeah, nowadays, climate change and habitat loss are leading to just notable biodiversity decline, which has an impact in the species decline as well. So what we can do just, and the response of that is one is genetic rescue. Genetic rescue is just supplementing individuals that are genetically healthy individuals into the populations that are suffering. And here it's more about just to rescue, as we can see, it's before they cross the line. If they cross the line, that's the extinction is another new area of genetic rescue research area. Now, what I were trying is before they become extinct, how we can just save them, how we can reverse before they extinct. And genetic rescue has benefits and risks. And the benefits are when the introduced individuals and the residents met and produce a fetal hybrid. Like this is observed in these two species in Florida panther and pigeon possum. In Florida panther, their name was dwindling to like 30 individuals, but now it's improved to like 270. And the negative outcome of the genetic rescue is when the introduced individual and the resident individuals produce less fitter than their parents. And this is observed in isolated wolf. In the isolated wolf, just I tried to put them up. The population are first established by two individuals, just one male and one female wolves that cross from the Ontario, and they were involving, and they got up to 25, but because they are closely related and they were inbreed. But after some time, one individual crossed the Lake Superior and from the North America, that's Minnesota, and joined the ice royal population, and it was very fitter than the inbreed population in the ice royal, and become related with everyone in the population in very quick, just in 2.5 generations. And after that, the population declined to only 2. So that's the negative outcome of the genetic rescue. So what are the so far the evidence of genetic rescue? It's very hard to do the experimental study on genetic rescue because it's hard to track how they met in fields. But the option is just doing simulations, and the available evidences in simulations are mixed. Some are in favor of it. Just they are saying just it's good, and we have to adapt and we have to apply it. And some are opposite. Is it has an intended consequence, like Chris and Texas and Hobart. This is what really motivates us to research or to do simulation on genetic rescue. And when you come to our simulation approach, is just we have two population. One is the insurance population. The insurance population is just without disease and evolving naturally. And the other is wild population that are suffering due to disease, that this is infectious disease. And in the wild population, what we assume is just the population will start adapting the disease just against the disease. So when we supplement individuals from the insurance population that's disease-free to the wild population, how it will help is that it will decrease the fitness of individuals that are conferring mutations that help them to mitigate the disease, or it will improve the genetic diversity of the population and improve also the overall health of the population. These are the questions that we want to tackle. Okay. And our example is just Tasmanian devils. There is a recent document that tells that Tasmanian devils are evolving against the disease. The model we applied is the non-rice fisher model. And this is the flowchart of the simulation. We first have the burn-in parade, and we have just we and the disease introduced in the after the burn-in parade, and we create the insurance population, and we disease spread, and and after 5 years post the disease, we supplement the individuals and we add them to the gene pool, and they reproduce adult. Yeah, this is the preliminary result. This is the scenario we have, like when we do nothing, when we apply 10 individuals, 14 individuals, 40 and 56. I picked for 14 individuals. This is when the disease transmission is low, and when the beneficial mutation that helps the wild population to evolve against the disease just to live longer. We didn't observe the extinction, and the first graph is just when the population just the individuals with the proportion of infected individuals. Bimodal, the lower one is that suffered due to the disease, and the upper one is that cleared the disease. This is for the medium, and this is when the disease transmission is high. The extinction is, the simulation that went extinct are 67.6. Yeah, and this is the heat map. Maybe I'm running out of time. Maybe I will summarize. Thank you so much.
Thanks, Hub 2. Sorry you had to be cut off. That was really interesting work. I'm not sure if we have time for questions. We can ask one. Does anyone in the Hobart Hub have a question?
Sorry, just a quick question. So is the Maynard Jones there? I think she's important.
Okay. We just consult her to choose the parameters that we are incorporating into our model. That's why I acknowledge her. Okay, thank you.
And there's one question on Slack, apparently. So this question is for Amon in Christchurch, by Hakim, who asked how do you validate / measure the performance of the clustering approaches?
Thanks, Hakim. I was actually mid-typing a response to that, but I'll just answer in person. So there's no ground truth data to use to evaluate your clustering approach, which makes it a bit difficult. One simple approach we rely on a lot of the time is sampling your resulting clusters and then manually reviewing them, so you mark them as accurate or inaccurate. This is helpful, but it's still not perfect, because if manual review is not perfect, sometimes you can make mistakes or different reviewers disagree on what the outcome should be. So we also mark them as maybe sometimes, and we just discard those. That's basically it. Sometimes we stratify as well by the type of cluster you have. So whether it's a simple 1 ID from dataset A and 1 ID from dataset B versus a 1-2 cluster or a 2-2 cluster, that type of thing. Yeah, thank you.